Afternoon, everybody. A very warm welcome to the Integrative Environmental Health Sciences Seminar Series. This binary series welcomes members and guests of both the P30 Center for Urban Responses to Environmental Stressors, CURES, and the P42 Center for Leadership in Environmental Awareness and Research, CLEAR, both funded by the National Institute of Environmental Health Sciences. I'm Graham Parker, the leader of the Translational Research Support Corps of CURES, and it is my great pleasure today to introduce our guest speaker and CURES researcher on the Environmental Epidemiology and Pediatric Immunological Development Research Interest Group, Dr. Cathy Peltz, co-investigator and operational director for the Trans-America Consortium for Health at the Henry Ford Health Department of Public Health Sciences. Cathy has developed a strong background in orthopedic research and epidemiology, conducting orthopedic research in the Bone and Joint Center at Henry Ford Health before joining the Department of Public Health Sciences to oversee the All of Us research program, focusing on precision medicine and patient-centered outcomes research. Cathy completed a Bachelor of Science in Mechanical Engineering at Michigan State University before pursuing a PhD in bioengineering at UPenn. This was followed by postgrad training in epidemiology at the University of Michigan, Ann Arbor, before accepting a position as an assistant scientist in public health sciences at Henry Ford Health. In addition to Cathy's ongoing funding to engage, enroll, and retain diverse participants into the All of Us research program, Cathy was a recent recipient of CURES pilot project funding to examine lead exposure as measured in baby teeth. But today's presentation will focus on the history of the All of Us research program, detailing recruitment, current status, and focus, and an overview of the All of Us Researcher Workbench. Cathy will explain the data currently available to researchers, examples of current funded projects, and how CURES and CLEAR researchers can get involved. Cathy Peltz, the All of Us research program, current status, and data available.
Okay. Thank you, Graham. Can everyone hear me okay? I thank you all for having me. I'm very excited to talk to you all about this. And as I said before some of you joined, I'm actually coming from the All of Us offices in Bethesda, Maryland, because I'm here for a meeting. So it's very coincidental that I'm here today. So Graham gave a great kind of teed up what I'm going to talk about. So as he said, I'm one of the MPIs for our award for the All of Us research program. The other PIs are Chris Johnson, who a lot of you know pretty well because she is also very much involved in CURES, one of the co-PIs, and then Dr. Brian Amadani, who is in the Center for Health Policy and Health Services Research here at Henry Ford. Our award we've had since 2016, and really it is mostly to enroll and retain people into this and help build the resource. We lead a consortium of other sites doing the same thing that is based in our pre-existing collaborations within the Healthcare Systems Research Network. Those are people like Baylor, Scott and White, Essentia Health. There's 7 of us throughout the country. And then there are also a number of other awards from all of us for many different reasons. There's an award to build the portal that people log into. There's an award that curates the data. There's an award that engages participants. And then there are a number of awards that are called HPOPI awards, like we have here. And those include places like Harvard, MGH, Arizona, a lot of academic medical centers. So we're in really good company. So just got an outline of what I'm going to talk about today, just give you some background on the All of Us research program. And then I'm going to talk a little bit about the data that's currently available on the Researcher Workbench. There are different tiers to the Researcher Workbench. So some of that data is available for anyone to just go on and look at, and then some of it requires some more, and we'll talk a little bit about that. We'll talk about some current projects that are using the AOU data, and then we'll talk a little bit about how you can get involved in things that you might want to do. So the All of Us research program, again, is a historical longitudinal effort to gather data from at least 1 million people. It takes into account individual differences in lifestyle, socioeconomics, environment, and biology. And it's really about precision medicine. It's part of the broader precision medicine initiative, actually from President Obama when he was in office back several iterations ago. But the mission is to really accelerate health research, enabling individualized prevention, treatment, and care for all of us. And it kind of has these three intersecting sub-missions or objectives. And one is to nurture partnerships for decades. This is intended to be a very longitudinal cohort with 1 million or more participants who reflect the diversity of the United States and its territories. The second is to use those partnerships to deliver one of the largest, richest biomedical data sets that is available and secure. And then using that data set to catalyze an ecosystem of researchers, communities, funders who use that data for their research. And all of this is made possible by a team that maintains a culture. The program has a number of core values that I'm not going to go into today, but those are available on their website. So again, just very, very high 300,000-foot view. We engage people in communities, many of whom have been left out of medical research in the past. We combine biological factors and social determinants on a large scale, and then we follow these participants as they move and age and grow. This is just a very kind of high-level timeline of the program since its inception. So again, the Precision Medicine Initiative was announced in 2015 in the State of the Union, and then in 2016 is when the first applications started being announced to become a partner in this study. We were one of the beta testers in 2017. We launched in 2017, and then in 2018, the program launched nationally. And a lot of this further timeline is about the data browser being launched, which the data browser, we'll talk about what that is in a little bit. Then the Researcher Workbench, when whole genome sequencing and genomic data began to be integrated, and when that became available to researchers. And then really where we are now, there's over 10,000 researchers using the data. We've launched pediatric enrollment and kind of what things are happening right now. So again, we are inviting over 1 million people. Currently, there are about 875,000 people enrolled. That means consented at any part. There are almost 600,000 people that have completed all initial steps of the program, and we'll talk about what those initial steps are in a few minutes. And then there's over 600,000 people that have some sort of data available in the Workbench. This is from the V8 data release, which was from February of 2025. They're scheduled to be a V9 release this year, so that number will just increase and the types of data will just increase. So as I said, this is over 600,000 people have data available in the Workbench, and about 84% of those people self-identify as members of communities underrepresented in biomedical research. And that can mean a lot of different things. There's a lot of different categories for what we call UBR. It's income, it's education, it is race and ethnicity, it is sexual and gender minority, it is education. So there are a lot of things that can make one a member of an underrepresented group. So currently, this is how you enroll into the program and the kind of things that you are asked to do. I will say that you can choose to do, if you enroll into the program, you can choose to do any or all of these. You don't have to do anything other than consent. But you'll be asked to answer surveys and things like that. So first, you're enroll into the program. Currently, I will say we are broadly recruiting 18 and older. It says plan to include children in the future. That's because the pediatrics is still in a kind of a beta phase that is scheduled to be largely rolled out later this year. But Henry Ford is one of the pilot sites for pediatrics. So we are currently recruiting children 0 to 4. The consent is online, it is interactive, and then there's also a second consent, which includes authorization to share your EHR data with the program, which is then in the Researcher Workbench, which we'll talk about later. You then are asked to answer a number of surveys. There are 6 that are available at the time that you enroll, and then others will be made available as they are released and announced. We haven't had a new one in a while, but there is one about to be launched this year. We can talk about that in a little bit. And then you can provide physical measurements. There are a couple different ways to do this. Right now, we are only enrolling people in person who are part of a partnered research study or a parent of a pediatric participant. So you can participate completely online, and you can self-report height and weight. That's the only thing that is available to be done by yourself, self-reported online is just height and weight. But if you did a clinic visit at one of the recruitment sites, you would also do blood pressure, heart rate, BMI, hip and waist circumference. And then also you could provide a biosample in person. That would be blood or saliva and urine. You can also still provide a biosample remotely by, you can request a saliva kit that will be mailed to your house and you mail it back. You can also choose to share data from wearables or digital apps, including like Apple Health or your Fitbit, things like that. And that data from the Fitbit, at least, is in the Workbench as well. So that's just a very brief overview of the program. If you have more questions, you can contact me or answer. I'm happy to answer at the end of the program. But I really wanted to talk mostly about the data that is available and just to kind of as an advertisement, it's open and available for people to use. So how does the data get to where it is? So first of all, the participants agreed to share their data through multiple sources. As we said, it can be survey data, EHR data, physical measurement data. The Data and Research Center, which is at Vanderbilt University, then securely processes this data and then shares this data aggregated in the data browser or individual level in the Researcher Workbench. Researchers can then register for access to the Workbench and then create projects in the Workbench and then publish or share that information. We'll talk about this in a little bit, but it is important to note that the Researcher Workbench is completely a cloud environment. You cannot download data from it. All your work is done in the cloud environment. So this is just a kind of high level of the data curation process. I'm not going to go into detail, but again, as we said, data comes from multiple sources. The data is then harmonized. The EHR data is harmonized in the OMOP model. And then all of us, the DRC, we call it, the Data and Research Center, cleans and processes the data. They then make it available in a curated data repository. And then there's also a data dictionary that documents what data is available. So data types, this is currently, as of the release of February 2025, what is available in the Workbench at different levels, and we'll talk about that in a little bit. So there are over 600,000 people with survey responses, over 500,000 people with physical measurements, almost 450,000 people with genotyping arrays, over 400,000 with whole genome sequencing, almost 400,000 with electronic health record data, and then, almost 100,000 with some different types of genomic data, structural variants, and then, just almost 3,000 with long read whole genome sequencing. There's also almost 60,000 people that have Fitbit data, and then 36,000 people that have Exploring the Mind text. So Exploring the Mind was one of these partnered research studies, which is a supplementary study to all of us, where someone wants to ask additional questions, and then that data goes into the Workbench as well. So these were kind of some like cognitive tasks that people were asked to do, and this last release of the data was the first time that data became available to researchers. And then again, this is just an example of, obviously, when you want to combine the different types of data that you're interested in, that those denominators are going to change, right? So say you wanted to do a study that included whole genome sequencing, short read, and survey responses. Well, you'd have about 400,000. But see down at the end, if you wanted to do a study that had Fitbit, EHR, and survey responses, and whole genome sequencing, you'd only have about 30,000. So again, dependent on the types of data that you're interested in, your numbers can be very high or a little bit lower. But in general, the numbers are very high compared to most of the studies that we do. Okay, so we're just going to go through the types of data that are available, and then kind of some of the advantages and limitations of the types of data. So for electronic health records, we have conditions, we have labs and measurements, medications and procedures. The advantages to that are the data is available as far back as the 1980s for some participants, pretty much as long as health systems have had electronic health records, and it is standardized to the OMOP model, so it is very easy to compare across participants. However, some of the limitations are we don't currently have clinical notes or imaging, even although that is on the kind of roadmap for something they want to incorporate. Again, not all participants have electronic health records available. You can participate and choose not to share your electronic health record. You can also participate and agree to share your electronic health record, but you did not participate. You may have not participated through an institution that is able to share your electronic health record. So if you signed up through Henry Ford, enrolled through Henry Ford, are a Henry Ford patient, we are sharing your electronic health record, and you consented to share we are sharing your electronic health record. There's another kind of avenue to enroll in the program that we call direct volunteer. So if you live, say, in Wyoming, where there isn't a health provider organization that is funded to enroll people into the study, you can still participate in the study. You can complete all your surveys online. You can do a mail-in saliva kit. You can complete the consent to say you're willing to share your electronic health record, but your provider is not then funded to share your electronic health record. There are ways around that we are working on, and there's a whole award that we're going to talk about later called CLAD, the Center for Data Linkages, that is trying to figure out ways to incorporate the data and to receive the data for people that agreed to share it, and their institution has a way to share it. Participant surveys. So these are listed, all the surveys that over time have happened. The basics is just kind of a demographic survey. Lifestyle is really about substance use, overall health, personal health and family history, healthcare access and utilization. And then those were the kind of the baseline surveys that were available for most of the time. And then over the years, they added a social determinants of health survey, two kind of behavioral health surveys, emotional health, history and well-being, behavioral health and personality. And then we had two surveys during COVID that are now closed and not available to new participants, but that data is on the workbench as well. So it's interesting to note too that when we say that 600,000 people have survey data available, that doesn't mean they have all of this survey data available. So most people have the basics. You have to complete the basics before you do anything else. But, for instance, if I enrolled in 2017, which I did, and the social determinants of health survey was not available at that point, I'm asked to complete it later, but not all participants obviously respond to longitudinal asks. So as the surveys kind of get added on, you have the number of people that completed them are less. So as I said in limitations, some surveys are completely skipped, and then questions are also able to be skipped, or there's a prefer not to answer opportunity for some of the questions in the surveys. There are also disparities in who skips the surveys, and there are high missingness in some demographics, so that can affect the data that you are able to access. So for physical measurements, like I mentioned earlier, we have blood pressure, heart rate, height, weight, BMI, hip and waist circumference. And so the advantages of this is that you have kind of some different sources of this data. You can get some of this data from electronic health records. You can also get this data from the in-person baseline visit if they completed an in-person appointment. And then they also can self-report height and weight if they are a remote participant. And then again, this can be combined with additional measurements from electronic health data. So again, if you have just height and weight, then you can, compute BMI or something like that. The limitations are the units must be normalized because, different institutions use different units. As I mentioned before, self-reported measurements include only height and weight. And then obviously, if you have access to self-reported height and weight from an in-person visit or from electronic health record, they may not be the same, and then you have to decide which one to use. Fitbit data, again, they have a lot of data. They have heart rate, sleep data, step counts, device type, all of that. The advantages is this is longitudinal Fitbit data, and some people it goes back as far as 2009. They did have a pilot study where they offered Fitbits to some participants. It was called the Wear study. So there are people that had their own Fitbit and signed it up, and then there are also people that received a Fitbit through this Wear study, and that was targeting people that were UBR representing underrepresented groups. So there is some diversity in the Fitbit data. It can be combined with other data types like survey data or genomic or EHR data. Some of the limitations is that it's not raw data. It comes from Fitbit. It's currently only Fitbit devices, so you can't get wearable step counts and things like that from devices other than Fitbit currently. It's the cohort with Fitbit data is less diverse than the overall all of us count cohort. The monitoring period for people that had a device versus received them through the Wear study are not always going to be the same. And again, some of the users applied Fitbit data like nutrition are not available. Okay, so genomic data is a type of data that I am less familiar with, so please don't ask me detailed questions about genomics. But here we go. So they have genotyping arrays, short read whole genome sequences, structural variants, and long read whole genome sequences. So the advantage, the best advantage, obviously, is this is I don't even know if we need to say one of anymore. I think it is the largest and most diverse set of whole genome sequences that is available for research. However, there are some limitations to people that are interested in genomic research. There are no gene expression or protein levels. Some phenotypes may have limited availability. Again, not all participants have genomic data available. And working with genomic data can incur large costs. And we'll talk about when we talk about how to access the data, what the cost is to do this analysis in the cloud. So as I mentioned earlier, there is an awardee within all of us called CLAD, the Center for Linkage and Acquisition of Data. So they are working on pulling new data into the workbench from existing data sets like mortality data, claims data, geocodes, environmental data. I know certainly of interest to this group, and then data from health information exchanges, which is what I was talking about earlier of people that agreed to share their electronic health record information, but were not associated with an institution that was funded to provide that. That's a way that they can get electronic health record information from those people and people that have data through their institution because it may be from another institution that they also got care from that is not funded. So there are going to be a lot of slides coming up that have QR codes on them. I will work with Lynn or whoever I need to work with to send these slides out so you don't, and if you don't catch all the QR codes, you can look at them afterwards. But this QR code and link here go to the data roadmap. So there is a long-term strategic plan of what kind of new data that we hope to add to the workbench and when that is going to happen. And then it does allow you kind of plan ahead or think about the kinds of study questions you would like to answer and to kind of pre-plan your study, right? When this data becomes available, you're ready to go. You have your study ready. Okay, so now I'm going to talk a little bit about how to actually use the workbench and what it is like. So first of all, there is something called the data browser. So there's a lot of links and QR codes here, but if you just go to researchallofus.org, it kind of has all of this living in one space or links to all of this. So the data browser is really mostly aggregate data and summary statistics, and it does have information from electronic health records. It has data from genetic variants, survey responses, and physical measurements. However, it does not have any personal identifiers, and they are not allowed to report data counts fewer than 20. So if you're looking at a really rare disease, you may not, it will just say less than 20. It might be 1, it might be 19, but it's less than 20. And then again, it's only summary demographic information. It also does not allow you to cross tabulate. So you can look up, say you're just doing preparatory to research and you're like, I want to see how many people in the cohort have asthma. You can look that up, but you can't find out how many women aged 18 to 25 have asthma, right? So you can't kind of cross tabulate. And that kind of work you would need to actually register and use the Researcher Workbench. So I'm just going to go through a few screenshots here of what the data browser looks like. So this is what the data browser looks like. It has a box for a keyword search on top, and then it has kind of all the different data types that it would pull from on the bottom. And then even without keyword searching, you can just kind of look at things like here's top 10 conditions by participant counts. So pain is the top, metabolic disease, mass. Again, these are very, very general, but this is just kind of the top 10 things that come up from the EHR data. So, and then you can also look at each survey and you can look at the entire survey and all the questions and all the branching logic. And then it shows you, see in the numbers here, like how many people completed that. I'm sorry, that's not how many people completed that question. That's way over 1 million. but you can see what the choices are, and you can see all of that from each survey. And you can also download the survey here. So if you're interested to see if a certain question was answered, if there's something you're looking for, you can look at that, each of the surveys here in the data browser. And then you can look at, there is some demographic breakdown. It's only certain, so there is sex, and it's just female, male, and other. It also has age in 10-year increments. So you can get a little bit of an idea, but again, this is very kind of preparatory to research, like, are there enough people with my disease? Are there enough people in the age group that I'm looking for in my disease? But again, as I said, you don't know how many females or males are in each of those age groups, and there's not a lot of cross tabulation. It also shows you what SNOMED codes, these and ICD-9 codes and ICD-10 codes that this was pulled from. So you can see, overall, sorry, this is really, really small, overall pain has, almost 300,000, but then it shows where that comes from. Acute pain was so many, chronic pain was so many, rest pain, so it comes from a number of codes in the EHR to add up to the overall pain number. And then this again is just, you can do some searching for genetic variants within the data browser, again, high level aggregate, and then you can drill down into specific variants and ancestry associations. But to get more detailed, you have to register for the entire workbench. So when I say the entire workbench, this is what I mean. So there's the public tier, which we just talked about mostly is the data browser. It also includes the Survey Explorer and the Research Projects Directory. There is also a publication directory now, so that has all the publications that have come from the workbench. The Research Projects Directory is helpful and interesting, but there are some limitations to it. So what the Research Projects Directory is, when you register for the workbench and you want to ask a question, you have to fill out some information about the question you want to ask before you even get to the data. So some of the projects in the Research Projects Directory, not for some, for any of them, you don't know if they've gone on or not. So say you were doing some preparatory to research work and you realized once you got in there that what you needed wasn't in there and you didn't go any further, that project will still show up on the Research Project Directory. So it's kind of hard to tell the stage that the project is in. However, because that data is available and who owns it and your institution and everything is in that publicly available information, you can contact someone and you can say, hey, I'm really interested in the study that you're doing. Can you tell me more about it? And they can say, oh, we're going to publish it next year or next week, or we didn't move forward with that project. It also is really trying to foster team science in that you can reach out to someone and say, hey, I'm doing really similar work to what you're doing. Do you want to work together? They don't kind of do that on the back end. So if you propose to do something that someone else is already doing, it doesn't alert you to that. It doesn't say, oh, we're not going to approve that because someone else is already doing that. But you can look and kind of see what's already being done and if your question is novel or not. And then there are 2 tiers of data once you register for the workbench and look at individual data, and that is the registered tier and the control tier. So the registered tier includes surveys, EHR data, physical measurements, and wearables. It does have some information stripped, like zip code is only at 3-digit zip code, and things like that. And then there's the controlled tier where the genetic information lies, and there are some more granular demographic data, like I was saying, and there are real dates, there are shifted dates in the registered tier. This is all explained to you when you get in there, but this is just a general overview of that. So as I mentioned, the Researcher Workbench is a cloud-based platform. It uses analytical tools like RStudio, SAS, and Jupyter. It does have graphical tools for selection and dataset building. It's a passport access model, so you can register if your institution has signed an agreement, and Wayne State has signed an agreement with that exists, and they have controlled and registered tier access. So if you have a Wayne State email, you can sign up for the workbench. The same is true at Henry Ford. And again, in this level, there is no separate IRB approval needed because the data is de-identified. So again, some really high level advantages and limitations of the researcher workbench. So again, it is unparalleled, these different types of data to be able to be looked at together. You have genes, you have environment, you have life circumstances, you have EHR surveys, and a lot of people. So there's a lot of information there. It supports replicability. So a lot of when the workbench was first released, they asked those of us within the program to do what they call demonstration studies, to basically recreate studies that were already out there using this data to show its utility. It's available to anyone with a secure internet connection. It has advanced computing potential, like I mentioned, R, Python, and SAS, and then it has some other tools that are specific to genomic analysis. And then it also provides the opportunity to work with each researchers from other disciplines and other institutions because of the cloud environment. However, like anything, it does have some limitations. It's not a representative sample of the United States. For instance, one of the demonstration projects that I personally did was on multiple sclerosis, and we found a much higher prevalence of multiple sclerosis in the All of Us data set than in the United States. It's not really surprising. I think people with rare diseases and people that are sick, even though anyone is open to participate in All of Us, people that have problems are more familiar with research, more willing to do research. They want to be a part of it. There is a learning curve with the data analysis tools. You have to be proficient in either in Jupyter Notebook, RStudio, or SAS, and you have to have the ability to code in R, Python, or SAS. You get an initial credit of $300 in computational credits. And it says here that most researchers will exceed that, and then you have to set up a Google billing account. For genomic analysis, this is 100% true that $300 goes really quickly. For instance, the multiple sclerosis study that I was talking about that we did was a purely kind of epidemiological description paper. We did not exceed $300 credit just doing that one study. So it's really dependent on how involved your study is. So current research, I'm just going to go through some high level examples of some things that are and have been done using this resource. So there's over 22,000 projects in the Researcher Workbench, but as I noted, it's hard to tell how many of those are active projects and maybe how many of them may have been abandoned at some point. There are over 1,100 publications in peer-reviewed journals now, which is really amazing and interesting. I used to have a slide, but I have not had the chance to update it in the last couple of years, that was showing kind of the comparison to the UK Biobank. And the UK Biobank has many more, obviously, publications than that now, but it took them longer to get started. So our trajectory kind of started earlier. So I think there's a lot of hope that it will just continue to grow exponentially. The top 5 conditions being studied in the workbench, again, this is based on research, the project directory. So again, some of them may be abandoned. Cancer, cardiovascular disease, diabetes, mental health, and hypertension are the top 5 conditions being studied. So these are these are very high level slides, and the program has some people that kind of pull these. They call them research highlights. They're available to be shared with participants. They're very at a community kind of level. But also to share with researchers kind of the utility of the study. So these are all published peer-reviewed publications. So one of the studies used EHR. It was about 250,000 All of Us participants. They found some pairs of medications that could cause health problems if they were taken together, and these were unsafe combinations that were not known before. So it was really again, the thing we always want to say here, and it's hard for participants because a lot of times a participant sees something like this and wants to go to their doctor and say, see, I shouldn't be taking this medicine, or can you check my medicine again? These are research results. There's a lot more research that is needed, but there have been things already in All of Us that have been used clinically. So in a very short time period, the Researcher Workbench is only about 5 years old. So This is a study about using AI for drug discovery, so very timely. This researchers in this study again used EHR. Almost 250 LLS participants. What they did is they used ChatGPT to help researchers find 10 existing medicines that might be useful in Alzheimer's disease. And then they used the data from all of us to look at those 10 drugs. And then they found that 3 of those 10 were worth studying further. So that kind of shows how this kind of large data can help researchers find better promising medicines faster than before to kind of get into clinical trials. This was a really interesting study. Again, this was based on genomic data, but it was a specific drug that was being used to treat cancer. And basically there's 2 doses of this drug that are very dependent on a gene. So they test the genes. And the genetic testing is common, but what they found in this study that there was a really rare finding. Again, it's 14 people. It's not world changing, but it's world changing for those 14 people that the test wasn't working as noted, and they needed a more delicate test to be able to tell if they had this genetic thing that meant the cancer drug wasn't working. So again, super rare finding, but impossible without the number and the power of the data, for the breadth and depth of the data in all of us. This is a study that used survey data, so we're just trying to show different surveys. So this is kind of using, we mentioned they had a couple of studies that were done during COVID specifically. So they were finding about social support and feelings of depression. And basically, not surprisingly, they found that social support was related was helpful for people that were most at risk for depression. But it was unique because this data, one of the beauties of all of us is that they were able to build and deploy this survey to a waiting number of hundreds of thousands of people very quickly during the COVID pandemic. And so that's kind of the idea, is that in the future we'll be able to kind of add surveys as needed in general things. There's an environmental survey being built that's kind of on hold for budget reasons, but it responses to things that are happening in the world, to things that are happening in healthcare. It's kind of this group of people that are willing and ready to answer questions and complete surveys, to participate in follow-up studies. So it really helps things move faster. This is a study about dementia risk. So they basically found that high blood pressure it confirmed that high blood pressure was a risk factor for dementia. This was again using EHR data. So you see a lot of the studies are using EHR data, but I think what's powerful is when we're able to combine that with others. Like for instance, this study, it used EHR data, but it also used data from Fitbit. So it used data from people that had EHR and Fitbits. And they found that taking a certain number of steps a day was linked to a lower risk of type 2 diabetes than taking a lower number of steps every day. So, the Fitbit data being linked to EHR data is something that doesn't really exist in this kind of scale and other researchers. So this was a really unique combination of the 2 types of data. So again, if I have wet your whistle, if I had made you interested, these are the steps that you need to take to register for the workbench. Step 1, if you are affiliated with Wayne State, your institutional access has been confirmed. You create an account and verify your identity. Then you have to complete some training and sign a code of conduct. And then you're ready to go. Again, there's a number of QR codes here. Again, we'll send out the slides, but this just links to actually what the code of conduct is. You have to comply with a number of policies. There's a publication and presentation policy, a policy on stigmatizing research, and a policy on the ethical code of conduct. So like, for instance, you are also not allowed to report numbers less than 20. So if you're doing some study on rare diseases, you have to say less than 20. There's a lot of resources available through the workbench and through different partners of all of us. So there is a user support hub that has a lot of videos and tutorials and articles. And as a help desk, they support a weekly office hours that has like usually those office hours have a specific focus, but then also has time for open questions. And there's also a community forum where you can ask and answer questions of other users of the workbench. As I mentioned, the community forum. So you can share tips and get support. You do have to sign in with your credentials and be a registered researcher in order to use that. They also have live virtual events. Like I said, these office hours. There's also orientations for new users and drop-in office hours. And again, there are video tutorials. So how to use the genomics data, how to use R, how to use SAS, some great support, some great resources here. Billing and costs, as I mentioned earlier, you get $300 in credits when you register. Using the cohort and the dataset builder are free, and then you pay for cloud services, which is analysis, storage, and again, depending on the type of research you're doing, you can go through that $300 really quickly, or it can last a little bit. These are just some tips for minimizing your costs. So you can, pull less data, do a custom query, test your analysis on smaller data sets. And then once you've kind of optimized your workflow, you can build a larger data set. You can save files to your workspace for long-term storage, and then you can delete or pause environments when you're done with that. And so if you've built a cohort and you just leave it there, you continue to incur a little bit of cost. So you can delete it, but you obviously have to leave it while you're publishing until your thing, until it gets published and all of that, because you can't download the data. This is a new, relatively new kind of resource that exists. The University of Arizona is one of the HPO partners, but they also recently got a award to support researchers. So they have a consulting service, and you can get up to 15 hours of one-on-one consultation at no cost. So you can contact this service, and they can talk about feasibility. They can talk about doing some of this kind of preparatory to research work to kind of tell you if the data is even there. So this is a really, really great resource if you're kind of coming at this without a lot of background and support in being able to do the analysis yourself. You can use this resource for your first study. There's a newsletter called the Research Roundup that kind of talks about new studies that are coming out and where we are with the number of studies and all of that. It's pretty interesting. It's just an email if you want to sign up for that. And then this is just kind of a plug. If you're already doing work on the workbench, the deadline is tomorrow, so it doesn't give you a lot of time. You can submit an abstract to Science Day, but even if you're not already doing work, it's a virtual meeting, April 15th, all of us Science Day. It's just going to celebrate all of the research that's being done. There's going to be lightning talks, longer talks, virtual poster presentations. It's going to be really interesting to just kind of see what is happening in the workbench and the kind of science that is already coming out. And then I think this is one of my last slides. There is a specific page here for funding opportunities. There have been some specific ROIs, RFAs that have come out for all of us, like specifically using part of the RFA is to use all of us data. I haven't seen one yet, but Chris Johnson let me know that she was at a meeting and talked to some NIEHS folks. And they're really interested in using in the All of Us data and really using it for environmental data once that starts being added. So I'm really hoping that as hopefully, the funding landscape maybe gets a little bit more optimistic, that there'll be some more specific AOU specific funding opportunities, hopefully in the NIEHS space, but there have not been yet. And again, we always end our presentations by thanking our participants and the dedication of our researchers to enable these discoveries. So thank you. Happy to
That was
take
fabulous,
any
Cathy. I'm
questions.
sure there's going to be a lot of questions. Could I kick it off with asking a slightly potentially grumpy question? If somebody signed up to allow data from their electronic health record to be shared, is there a possibility that other people might know the results of a test before you do as a patient?
Um, Highly unlikely because it's not a um continuous process. We share that data once a quarter. Well, it used to be once a quarter. Now it's once a year because of budget cuts. And it only gets put into the workbench once a year. So it's delayed. It's not in real time. Um, but also it's completely de-identified. So you wouldn't be able to link it to a person. But unless someone never, received a result or they got a test that they never viewed the result or their provider never was able to contact them theoretically, but you wouldn't be able to link it. One of the things in the code of conduct is you have to agree to not to never try to re-identify a person um, based on their electronic health record. But
Yeah, it wasn't from the point of view of identifying the patient. It's just the patient's compliance and might change, shall we say.
yeah,
But
Cathy,
you're
they can't even tell if they're from Henry Ford, right?
no, we
You can't
can't
even
tell
tell what
what,
institution
what
people are
location
from.
they're from. No, when it's in the workbench. I mean, you
Alan,
can
you had your
guess
hand
based on
up
their
first.
location, but you don't know where the electronic health record is coming from. There's no like source.
Go ahead, Alan.
Yeah, I have two questions. First of all, thank you very much. It's a fabulous resource. And I apologize if I missed this, but regarding location, is the zip code of residence available?
The 3-digit zip code is currently available. The last 3 digits. I'm sorry, the first 3 digits. They are in the controlled tier. The um I think they're talking about adding full zip code, but to my understanding, it's currently just the first 3 digits of zip code.
Because when it comes to environmental studies,
100%.
location is absolutely
100%.
important.
Yes.
Second question is to help minimize the cost involved in data analysis. Could a user pull data and then export it and then use that locally for their, with their own
You
favorite
you
tools?
can't pull data down, you can't download or export data. It has to all be done in the cloud environment. Yeah.
All right, thanks.
Yep.
Paul, I think you want to ask a question, but you're muted.
Thank you very much, Graham. You were correct. If I could follow up on that. So you can't download the data, but you can download the results of your analysis of the data. Is that correct?
You can you can create figures from the and use them in a publication, but you cannot download yes, and you can download your results. And you can have those in a Excel, the summary statistics, but you can't download individual level data. Yes, you can download some. I see some summary statistics. Like for instance, for the paper, you can create a figure in the cloud, but you can also have your summary data and create your figure like in Excel, which is what we've done. So, by race, but and then you can that way get like you could have a cell that's 2 and a cell that's 5, but you're not allowed to report that you would know, but you can only report as less than 20. But yes, you can download your summary.
So let me ask a completely different question. And you have genomic data. Who generates that? Who does the actual analysis? And again, with that, I think you talked about having physical samples for where are those stored and are those accessible or just the survey data?
So right now, in the beginning, there were 3 awardees separate that did the genomic analysis. It was University of Washington, I think it was Baylor, and then the Broad Institute. And with budget cuts, it's now only one institution. And we're not doing, they're currently not offering new, the return of results to people is not happening, but they still are sequencing new data. So I think the only one now is the Broad Institute. So they are generating the genomic data. There are quite a number of biosamples stored at the Mayo Clinic is where they're stored. They have been used for one. I had some extra slides in case we had time, but we didn't have time for partnered research studies. You can find out about those on the website. So there is one study so far that has used those. It was actually an exposomic study through NIEHS that is looking at some things that has not, the results of that study haven't been released yet. But they're actually this year, it is on the agenda, it is on the goals for this year to have a RFA where people can request to use the samples for the first time. So you can, for the first time this year, I'm not sure if the deadline will be this year, it will come out this year, is the plan to request to use samples for your own study. And I'm not sure, they're still working on the part that has to be is how that's going to work, right? How you're going to get the samples, or are you going to have to contract with Mayo to do the analyses, or really what's going to happen. But that is definitely not currently available, but is on the immediate horizon.
And then I assume any analytical data would have to go back into
Yes.
the all of a system.
That is one of the caveats to any, and we call them partnered research studies, is that your results have to go back into the workbench to be accessible by other researchers. Yes.
All right, thank you.
Chris?
Did you want to mention that they're working on an environmental questionnaire?
Yeah, so for years, I think pre-COVID, priorities change and funding changes and things happen, right? So they've been working on a survey for an environmental survey for a number of years. What I have heard is it has not yet been deployed. What I have heard is that they are trying to work with NIEHS really to fund the implementation of that survey, right? Because it takes time and money for our program partners to build it and to deploy it. So I don't know of the timeline that they have for that. But that is something that they are working on. And to whoever asked the question about the zip code, I know again, I don't have a lot of visibility into the CLAD award, but I know geocoding is one of the things they're working on. And obviously you need full zip code for that. So I'm not sure at what point that data will become available, but there is a large interest in the people that have a vested interest in this data at NIH to add environmental questionnaires, data exposures, a lot of that.
Cathy, with the hope that researchers can play nicely internationally, is there any efforts to collaborate with the UK Biobank to try and increase the power of data sets?
That's an interesting question. So it is available to international researchers. You can register for the Workbench as an international researcher. I think that happened last year or the year before. I don't know of any specific kind of partnerships trying to be forged to merge the data together to use it, but I know people that have used both sets of data and kind of, compared and contrast or added something on. But to my knowledge, there isn't any partnership being formed. But yeah, there'd be a lot of power there for sure.
The floor is still open if anybody's got any burning questions. Cathy, obviously, you've given us a lot to think about and a lot of QR codes to try and scan. So hopefully Lynn will be able to.
Most of that, if you just go to researchallofus.org, it's all there somewhere. And it's a pretty nice and easily navigatable website. But yes, I will send the slides to Lynn and she can send them out.
That's fabulous. Lynn, do you have some closing remarks?
I have a question.
Oh, go ahead, Suresh. Sorry.
Hi, Cathy. Nice resources. So, if I have a interest on particular data set and like I missed the first part of your talk, so where do I go and get it specific? Let's say I want to look at heart failure or some specific disease. So, how do I navigate? Where do I start?
So the first thing you can do is you can go to the data browser and you can just type heart failure into the keyword search. And that will give you a very high level understanding of how many people. So I'm going to assume with heart failure, you're going to get hits in the survey and personal health and family history, because there's probably a question about heart failure or heart disease. And then you're also going to get hits from the EHR that is going to talk about the different conditions. And again, it's going to be very high level. You'll be able to see a female male breakdown. You'll be able to see an age breakdown. You'll be able to see where all the SNOMED and ICD-9 and 10 codes are coming from. But to get more granular than that, that's when you have to go into the Researcher Workbench. But you should be able to get some very high level information just about what's the number of people that are reporting and kind of the specific codes from the EHR that are coming from, if you're interested interested in something very specific through the data browser, which is open to anyone. But then after that is when you would have to sign up for the workbench and create a cohort inside the workbench.
So it may be common sense, but ignore my, novice. So if I'm going to propose a grant, can I use these data set as a resources that I'm going to work on? And like I understand that you cannot download the data. So, like, so if we identify something, we do something like usually in NIH grant or any grant you are going to send, then you have to specify where you are going to store your data and those kind of stuff. So I think since it's NIH, you have all established in that way.
Yeah, 100% you can use this data for grants. There are people that have funded grants that were specifically the RFA called to use all of us data. You can also use the all of us data. For the person that asked about using the samples, that's one of the things they're trying to work through is like if you want to use the samples for a grant, do you have to, have the promise of being able to use those samples before the grant gets funded. So there's several, you have the grant pathway and then you have the approval to use the samples pathway. So they're trying to work out kind of the timing and the timeline of how those things go together. But if you're just using EHR and survey data, 100% you can use this data for grants.
Okay. So do you have imaging data in the EHR?
Do we have what? I'm sorry. No, there's not
Imaging.
currently any imaging data, but it is on the roadmap.
Okay, got it.
Okay, Lynn, I think it's safe.
Okay, if there's no more questions at this time, just, thank you everyone for attending today, and heads up for next week, we're having another seminar on Zoom. So this one will also be fully virtual next Thursday on the 29th of January, where we're going to have Dr. Sheena Martinez from the University of Illinois, Urbana-Champaign. She will be discussing the title of her seminar will be Connecting Neighborhood and Children's Health. So please log on for that as well. Go to events.wayne.edu and you'll find the link there. Thank you very much, Dr. Peltz.
Thank you all for attending.
Yeah, that was great. I loved listening to that. And I'm excited to check out the websites.
Great. Thank you.
And thanks, Graham, for introducing and for moderating the questions. And we'll see you all next time.